Hello everyone. Today we are going to have a look at an interesting phenomenon in C. elegans, the internal hatching or worm bagging. In C. elegans, eggs are generally laid outside and hatching occurs outside the parental body. But in certain conditions, eggs are retained inside the body and hatching proceeds inside the parental body. In general, in C. elegans, eggs are fertilized internally in the parental body when a mature oocyte is ovulated and is transferred into the spermatheca, where it is fertilized either by self-sperm generated during the L4 larval stage or male sperm deposited after copulation. Post-fertilization in the young adult hermaphrodites, fertilized eggs develop inside the body for around approximately 2.5 hours and are laid into the environment, which then hatch after approximately 10 hours of further development. But in special conditions, if any fertilized egg is not laid outside within this approximate 12.5 hours, then it is forced to hatch internally, and the internally hatched larvae will begin feeding its parent from inside, ultimately killing the parent. This event is called internal hatching or matricidal hatching or endotokia matricida and is defined as intrauterine hatching, followed by larval development leading to the destruction of the parent by the larvae. This process is colloquially called as worm bagging. The term bagging because the parental worm undergoing this phenomenon gains an appearance of a worm bag. This form of cannibalism is quite common among the Rhabditida nematodes. Interestingly, this phenomenon is not only restricted to nematodes but also seen in other genuses. For example, Adaptylidium mites display internal hatching. Under normal conditions, when the worms are grown in laboratory conditions, internal hatching is very rare. However, in conditions when the worms are transferred from a solid medium to a liquid medium, for example the actinic medium, and in the presence of unfavorable environmental conditions like starvation, overcrowding, and exposure to toxic substances and pathogens including bacteria, the frequency of worms undergoing internal hatching increases. Why would this worm bagging occur in the first place? This could be an adaptive response to stress like starvation and shall enhance larval viability by promoting dower formation in nutrient-deprived conditions. Furthermore, adult stage hermaphrodites exposed to nutrient deprivation stop laying eggs and experience internal hatching. The paternal body acts as a food source for the hatched worms and can provide sufficient food under starvation conditions for the hatched larvae to reach the resistant dower larval stage. Then these larvae are released into the unfavorable environment as the parent paternal body starts degrading. And this released dowers on return of favorable conditions, these dowers return to the normal developmental process and grow into adults. Like all other processes in the worm, internal hatching is also modulated and regulated by genes and does not occur only in the wild-type worms. In C. elegans, this phenomenon of internal hatching has been extensively characterized applying genetic screens for mutant animals defective in egg laying. These studies show that formation and functional well-being of the vulva, the vulval muscles, and the HSN neurons which innervate the vulval muscles are essential for efficient egg laying. mutations influencing these systems cause egg retention and internal hatching. Aging is also an inducing factor of internal hatching. It should be noted that there is also an age-related degeneration of the reproductive system, including the egg-laying system in all animals including C. elegans. Reproductive aging in matured C. elegans hermaphrodite is characterized by a progressive age-related decline of the reproductive function. This reproductive aging in C. elegans initiates at approximately around day 5 of adulthood, leading to a complete seizure of reproduction altogether after around 10 to 14 days of adulthood. Studies have shown that frequency of internal hatching increases with increasing chronological age and occurs primarily in mated hermaphrodites that reproduce late in life. Internal hatching was also independent of the level of early and total progeny production and the duration of exposure to males, clearly indicating that it is not caused by use-dependent damage accumulation and may reflect intrinsic age-related degradation of the egg-laying system. Additionally, dietary restriction displays to delay matricidal hatching, indicating that nutritional status influences the age-related degeneration of egg-laying system. Significant is that the age-related degeneration of the C. elegans reproductive system may be relevant to other animal reproductive systems and provide a significant or a well-trustable model system that can elucidate human complications associated with giving birth. It is also proposed that internal hatching is a life history trait in C. elegans. Though internal hatching is initiated by stressful conditions, internal hatching is apparently reversible if the stress condition is removed before the adult has sustained significant lethal internal damage due to larval movement internally. Induction of internal hatching is indicative of signaling or a decisive process in the adult hermaphrodite in response to environmental stress or stimuli, particularly lack of food. Egg laying ceases shortly after such stress begins. Although earlier studies indicate that internal hatching is a result of defective reproduction or the reproductive system in older worms, other studies indicate that internal hatching is not restricted to old gravid adults. Most importantly, internal hatching is a means for adults to provide nutrition to the progeny in food-deprived conditions. This is an indication of existence of a altruistic behavior in C. elegans, which acts as a survival strategy. Signifying the potential of C. elegans to respond to stress and change its reproduction from ovipary to vivipary. In this regard, reproduction is the production of a new individual, whether or not the parent survives the process of reproduction. According to definitions, vivipary is said to be adopted when the young are nourished from the parental source, including provisions from the mother's own soma, either before birth or when the parent retains the developing egg inside the reproductive tract or body cavities and give birth to living offsprings. Among the invertebrates, vivipary is generally associated with unfavorable or harsh environments, and facultative vivipary also represents a pathway leading to survival enhancement in C. elegans life cycle. Vivipary is not an unheard phenomenon among the nematodes and is known to occur in the oviparous parasitic nematode Haemonchus, in the bacterial feeding radiated nematodes, and in other nematode species too. intrauterine birth, which results in death of the parent, is relatively common in rhabditid nematodes and is typical in Caenorhabditis, and has also been observed in Meidinema ali, Metacorbels, and C.elegans. Viviparity and matotrophy are phenomena of considerable biological interest, and some animal lineages have evolved a form of matotrophy in which embryo ingests maternal tissue, as is the case in worm C.elegans, as I already said. The phenotypic plasticity of the worms associated with stress is remarkable. The body contents of the parent favors the internal hatched progeny to achieve a larger body size to mold to dowers. Further, the maternal body acts as a shield. In addition to being a nutritional source, it also acts as a shield and protects the internally hatched progeny from the prevailing harsh environmental stress or stimuli. Strikingly, facultative vivipary causes a reduction in C. elegans adult lifespan. Evolutionary theories of aging in context to aging in different genders stipulate that longer-lived gender will be the one that experience significant lower mortality in nature. The comparative evolution of longer lifespan in males of C. elegans certainly may be a result of greater mortality experienced by the hermaphrodites because of internal hatching. So vivipary is an altruistic adult behavior and an inducible pathway that enables some larvae to reach the dower stage during unfavorable conditions, including starvation and stress. Dower and vivipary thus represent complementary history traits projecting as fitness enhancing adaptations depending on harsh environments and ephemeral resources available. Altogether, internal hatching happens in a wide range of stressful environments. Internal hatching occurs in longevity mutants too, no bias. Internal hatching is very much inducible. Internal hatching results in healthy progeny. Larvae from internal hatching are capable of entering the dower stage under the same conditions compared to their counterparts from normal externally laid eggs. Consuming the parental body contents, the internally hatched larvae are able to proceed to the dower stage even under complete starvation environment, the conditions under which larvae from externally laid eggs cannot attain dower stage. These research outcomes from the C. elegans research community significantly put forward C. elegans as a significant and a very good model system to explore functional and evolutionary questions on longevity associated with reproduction, developmental plasticity, parental investment in progeny, and associated questions haunting reproductive and evolutionary biologists for a long time. So, the application of this model has full potential to elucidate human complications, for example, complications associated with giving birth. Thank you.